Hi everybody, Professor H here. Welcome back to another video for Psychology 9, Developmental Psychology, Fall 2020. We are going to talk today about Chapter 2. This is the first of 3 videos that will cover Chapter 2. This is the first part, which talks about genetic influences on development. Let me go ahead and start a screen share. There we go. Okay, now we got a screen share going on. Hopefully you can see my PowerPoint. My allergy disclaimer, once again, we have a fire going up in Big Bear, so there's ashes raining down. I am very allergic, but so far so good. Hopefully we'll get through this. Okay, I'm going to put us in presentation mode and we're gonna get rolling. Let's get rolling, okay. Basically, in this part of the chapter, we're going to take a look at genetic basis for development, genes and environment, genes and individual development. We need to have a basic understanding of genetics. I'm talking very, very, very basic in order to understand how humans grow and develop. So I'm going to try to provide for you in the next few slides in this particular part of our chapter a basic understanding of how genes work. And this is very basic. If you're a biology major, you may roll your eyes, but otherwise, hopefully this will give you a good understanding. We're going to start talking about chromosomes. Chromosomes, all humans have 46 chromosomes. They are paired up. You'll get one from mom, one from dad. So you have 46 chromosomes in 23 pairs. These chromosomes are made up of DNA strands, and along these DNA strands are little segments called genes. And these genes, like you have a gene for blonde hair, a gene for brown hair. You might carry both of these genes, one from your dad, one from your mom. The exception, every cell in your body has 46 chromosomes, with the exception of the gametes. Gametes are your sex cells. For males, that would be the sperm. For females, that would be the ova or eggs. These particular cells have only 23 chromosomes. They're not paired up at all. There's just 23. The reason for this is because for a fertilized egg, when that happens, when that fertilization occurs, you have 23 from dad, 23 from mom. That makes up that 46 total, which is the 23 pairs. Get all the things here. Oops, okay. So this is a little bit of a little image that can help us understand about the chromosomes, the DNA, and the genes. Our chromosome is made up of long strands of DNA. The DNA is considered the building blocks of life. These genes are small specialized structures that contain instructions for proteins. For example, if this is my gene, this top one, if this is my gene for hair color, it's going to tell a cell that creates the hair color what hair color I'm going to have. It's very simplified, as I said, but this is basically how it works. A chromosome is made up of these strands of DNA, and our next image will help us see that a little bit better. This is an image from your book. This is the chemical structure of DNA. We're not worried about the chemical structure of DNA. That's a little too detailed. We're not going to get into that. What you do need to know is that these chromosomes, remember that humans have 46 chromosomes, are made up of these strands of DNA, and that along these strands are our different genes. And all of these things are located in the cell body. So inside of this cell is where these chromosomes are. This is an image of a set of human chromosomes. They are arranged in 23 pairs, so you'll see it's numbered to 22. Because the first 22 are pretty straightforward, where you're getting one from the male, one from the female. So they're automatically paired up like that. They are matched pairs. my hair color from my dad, my hair color from my mom. These are matched genes, matched chromosomes, genes along those chromosomes. So the chromosomes will have a lot of genes along them, just for simplicity's sake. The last set, as you'll see here on the bottom, are not necessarily paired because every female has an XX. They have two X chromosomes. Every male will have an X and a Y. There are exceptions, and we'll talk about those later. But for the most part, females have two X's, males have an X and a Y. When reproducing, a female can pass only an X on. She only has an X. So when I pass on and I have a son or a daughter, one of my X chromosomes is going to be passed on to my son or daughter. A male, when their sperm fertilizes and they are, their child is either going to have an X or a Y, and they are going to determine the sex of the baby. And we will go on and find out more about that. Two important terms you're going to need to know: genotype and phenotype. Genotype is your genetic makeup. It is the total of the individual's genes. In other words, along those DNA strands, those little segments are our individual genes. That's our genetic makeup. A phenotype is the actual characteristics that you see. That's what is seen or observed, and it can include a wide variety of things. For example, a phenotype could be to have brown hair. Like I have darker hair. So if I have a phenotype for dark hair, it's showing that I have dark hair. I happen to also know that I have a genotype that includes blonde hair. This blonde hair is inherited from my mom, who is blonde. And I know that I have this blonde gene because I have a blonde child. And this blonde child can only occur if I were carrying a recessive blonde gene, which we will talk about soon. There are different types of genes. There are dominant genes and there are recessive genes. So dominant genes normally express the characteristics. So just like you were, any other thing, if something's dominant, they're the one that wins. They're the gene that's going to get expressed. For me, the dominant gene is dark hair, because dark hair is a dominant gene, and I'm expressing that characteristic. I have that dominant gene of dark hair, and my phenotype, the way I'm expressing my hair color, is according to that dominant gene, which is dark hair. Recessive genes are not expressed, although it is part of the genotype. So even though I carry that gene for blonde hair, I don't have blonde hair, but I'm able to pass that gene for blonde hair on to a child. An allele is an alternate form of a gene. So I might have an allele for brown hair and an allele for blonde hair. So those are alternate forms of the gene. I have the gene for this color, a gene for that color. Those are alleles, different alleles. There's another type of relationship between genes, because remember, we have paired genes. So I have a dominant gene and a recessive gene. The dominant gene is going to be expressed in the phenotype, what you see. There's another relationship called dominant recessive inheritance. That's a pair of chromosomes containing one dominant and one recessive gene. This is just talking about the relationship between dominant and recessive genes. And we'll explain all this. It'll be explained pretty soon. This is kind of that relationship between the two. And there are other relationships that we're going to talk about as well. All right. Talking about our dominant recessive relationship with genes. Most traits that people have are more complicated than just having one dominant gene and one recessive genes. Most traits involve more than one specific gene in order to express that. But we're not worried about that in this class. I just want you to be aware of it. Most things are polygenic, meaning there's more than one gene involved. And that's just important for you to know. This is the basics of how dominant and recessive relationship can work. So let's say we have a mom that has dark hair. She inherited 2 dominant genes of dark hair right here. We're going to put that as a capital D to make it easier to understand. So for dark hair, we're going to say as a capital D. We got a gene from dad, gene from mom. Dad is blonde. So let's say the father of the children. So this is the mom of the children in the box. This is the dad of the children in the box, okay? Dad is blonde haired. In order to be blonde hair, you have to inherit 2 recessive genes for blonde. In order for a recessive gene to express and be seen in the phenotype, you have to have a copy of the recessive gene from mom, a copy of the recessive gene from dad, in order for that to express. If there is a dominant gene present in those pairs, that dominant gene is going to be the one that's expressed. So let's take a look at how this would work. Along this side, we have the 2 genes from dad. We have the 2 blonde genes right here, recessive, blonde. That's why they're lowercase. I'm putting recessive genes, recessive alleles as lowercase. Mom has dark hair, and she has these 2, these are the 2 genes that she inherited. So this child, we're going to take this B from dad, this blonde here, and this dark-haired from mom. So anytime there is a dominant gene present, the dominant gene is going to express. So this kid has dark hair. This kid also got a blonde and a dark-haired from mom, all of them. If you'll notice here, each kid got a blonde gene from dad. For him to have blonde hair, we know he has to have 2 recessive genes. So we know that all the kids are going to get that recessive blonde gene from him. However, none of them are going to be blonde because mom has dark hair, which is a dominant trait. So these kids are all going to have dark hair because she is genetically has 2 copies of this. Okay? And you're probably thinking, oh no, I know people with dark hair with blonde kids. Look at me, dark hair, blonde kid, right? How does that work? If I only had 2 genes for dark hair, I would have no chance of having a blonde kid. All right, so in our second example, our mom has dark hair. Her phenotype is dark hair. However, her genotype has a dominant dark hair gene and a recessive blonde hair gene. So she has the capital D here and the lowercase b. So this is for our recessive blonde gene. This is for our dominant dark-haired gene. Dad, once again, he's blonde. He's got 2 recessive blonde genes. That's all he can pass on because that's what he's got. So, oh my goodness. So we have the lowercase b here, which is the recessive blonde. We have the dominant dark hair. So this kid and this kid both have the recessive blonde and the dominant dark hair. So mom passed on this gene to the kid. So dad passed on this gene, mom passed on this gene, dad passed on this gene. These 2 kids are dark hair, but they can pass on the blonde gene because they have this in their genotype. Their phenotype is dark hair, their genotype has blonde in it, right? Now here, if mom happens to, in that pair that she give, in the one cell, the one gene she offers and the dad's gene, we can see here that this kid inherited the blonde, the gene for blonde hair from mom, and of course the blonde hair gene from dad. So this kid is blonde. Same thing with this. This kid received, inherited this blonde gene and this blonde gene. Both of these kids are blonde. And once again, when they have kids, all they will be able to do is pass on this blonde recessive gene. Their children aren't going to be blonde unless the mother has a gene for blonde hair also. Here's our third example. Our third example is we have 2 parents now that have dark hair. This is a mom with dark hair and a dad with dark hair. However, mom's phenotype is dark hair, but her genotype is including that recessive gene for blonde hair. Same thing with dad. He has a phenotype for blonde hair. His genotype includes that gene for blonde hair. So here we have that dominant gene for dark hair and the recessive gene for blonde. So let's see how this pans out. Okay, this kid one here, this child has dark hair and does not have a chance to pass on the recessive blonde gene because they don't have it. This kid has dark hair, got the dark hair gene from dad, the blonde hair gene for mom. So this kid has dark hair and can pass on the recessive gene. This kid and this kid kind of have the same thing. This kid got the blonde gene from dad and the dark hair from mom. Once again, dark hair and can pass on that recessive gene. Phenotype is dark hair, genotype includes blonde. And this kid inherited this blonde recessive gene from dad and a blonde recessive gene from mom. So this child is blonde and can only pass on the blonde gene. Okay, so this is an example. I kind of clicked off that quickly. This is an example from your book also. This graph is in, or this chart is in your book. This talks about other traits that are pretty much single gene dominant recessive inheritance that have that dominant and recessive relationship. Most things are polygenic, meaning they have more than one gene involved. But these things, such as curly hair, which is dominant, straight hair is a recessive gene. You would have to get two recessive straight hair genes. Dark hair is dominant, recessive is blonde. We already went through that. Dimples is dominant, no dimples is recessive. Normal hearing is dominant. Deafness, some forms are a recessive gene. There is genetic forms of deafness, and so on and so forth. There are certain things that are related to that single gene dominant recessive inheritance. Okay, just a quick little thing here. Once again, most traits are polygenic and have an interaction of multiple genes. There is another type of genetic relationship that we're going to talk about called incomplete dominance. Incomplete dominance is a phenotype influenced primarily but not exclusively by the dominant gene. What this means is the dominant gene is still dominant, but that recessive gene actually has an impact on development as well. It's not just completely dominated by the dominant gene, it has an impact. And that's what we're going to talk about next. So this particular example, we're going to talk about sickle cell anemia. Sickle cell anemia is a kind of a terrible disease. It affects mostly people with African heritage, people that come from areas that had like high malaria rates and such. These are areas, and in the Black communities, we see more sickle cell anemia. It is more common in our Black population, especially here in the United States. So that particular disorder causes your blood cells, instead of being little round platelets, it causes them to be sickle or moon shaped. So they're kind of pointy. And what happens in this disease is when people have bad flare-ups, these sickle-shaped blood cells get stuck in the capillaries, and they get stuck in different areas, causing people extreme pain in their joints, causing breathing difficulties because the blood isn't flowing through the lungs properly, causing kind of super bad mega arthritis type of pain because these cells, these sickle cells, cannot travel through these smaller vessels. So this particular disease is a genetic disorder, and it is very, very painful for people who experience this. So we're going to talk about this now. I think your book will also give you a description of sickle cell. How does somebody get sickle cell anemia? Well, once again, it is one of those inherited traits. We have a dominant, we have a recessive gene, but in the case of the recessive gene that creates sickle cell anemia, that recessive gene will have an impact even if there is a normal dominant gene. So the way that I've laid this out for you guys to make it more clear and easier to understand is to use the letter N for our normal cell, the uppercase N, and we're using the lowercase s for our gene that carries sickle cell. So let's see what happens. We have mom who does not carry the gene for sickle cell anemia. She has two normal cells for her, two normal genes for expressing her blood cells. Dad is a carrier for sickle cell. That means that he does have the normal cell, the normal gene for creating the blood cells, but he also has the recessive gene for sickle cell anemia. Because sickle cell anemia, the gene for that is a recessive gene, you would have to have that gene from both parents in order to have sickle cell anemia. Just like we talked about with blonde hair, the same thing goes for the recessive gene when we're talking about sickle cell anemia. The difference is when you have a recessive gene that is showing that has an impact as well, it's an incomplete dominance, what we see is an impact. So this dad, although he doesn't suffer from sickle cell anemia, he carries this gene. This gene means that some of his blood cells, not all of them, but a few of his blood cells have that sickle shape, but not enough to cause him to have sickle cell anemia. It will, however, make him resistant to malaria. So by carrying this recessive gene, he will be resistant to malaria. This is biologically makes sense from an evolutionary perspective because people who live in a lot of those African regions where there's tons of mosquitoes and tons of malaria, you would be protected from malaria by carrying this recessive gene, and you would not be expressing the disease. So let's take a look at these first kids. First kid is getting a normal gene from dad, normal gene from mom. This child does not have sickle cell anemia and is not a carrier for the disease. In other words, they are not having the genes to pass it down to their children. Let's look at this child. We got another normal one here, normal one here. This child, no sickle cell anemia, is not a carrier, cannot pass it down. These two kids, on the other hand, they're getting this, they happen to inherit, by roll of the dice, they happen to inherit this sickle cell gene and from dad and a normal one from mom. So just like the dad, these two kids have resistance to malaria because they carry that gene. They do not have sickle cell anemia, although they will produce a certain amount of sickle-shaped cells. And they are both carriers for the disease. In other words, they can pass this on to their children, this particular gene. So 50% of these kids have resistance to malaria. These two do not. So in our second example, mom has one normal gene and one of the genes for sickle cell anemia, this recessive gene. Dad, same thing, same dad. We have the normal gene here and we have a gene for sickle cell here, which is a recessive gene. This first child, again, has inherited two normal genes that do not have the sickle cell trait. So this child doesn't have the disease, is not a carrier, cannot pass it on. This child, however, inherited the normal gene from dad and the sickle cell gene from mom. They are not having sickle cell, they are a carrier, they can pass it down, and they are resistant to malaria. This child is the same diagonally here. Same thing, got the, it's just that they happen to inherit the sickle cell gene from dad, not mom. They got a normal gene from mom. So once again, no sickle cell. They are a carrier, can pass it down, and they're resistant to malaria. This last child happened to inherit the sickle cell gene from dad and the sickle cell gene from mom. This child will have sickle cell anemia, and they will have that, that issue with their blood cells and have the actual disease. They'll also be resistant to malaria, but they have sickle cell anemia. This is one of those diseases that's kind of interesting from an evolutionary perspective, like why hasn't this gone away? Why is this disease still prevalent? And it does give resistance to malaria. All right, so here's our third example. Our third example for sickle cell anemia is that mom has one dominant normal gene and one recessive gene, and dad has sickle cell anemia. So he has two recessive genes that both carry the sickle cell gene. Okay, these are both the recessive genes for sickle cell, and he carries them. He has the disease, so he has, that's all he can pass on. So this kid has the sickle cell gene from dad, the normal one from mom. Once again, they don't have the disease, they're a carrier, they're resistant to malaria. Same thing with this kid. Got the normal gene from mom, sickle cell from dad. However, these other two kids, this one inherited the sickle cell gene from dad and a sickle cell gene from mom. Same thing here. This one got a sickle cell gene from dad and a sickle cell gene from mom. So both of these kids have the disease and are resistant to malaria. Now, in these cases, sometimes people who have this running in their family, sometimes they get genetic testing before they have kids so they can kind of know what their odds are of having kids with sickle cell. There's genetic counselors that people can go to talk about this and weigh out what they want to do, what their options are. And there are many other diseases like this, not just sickle cell, that other populations have. And genetic counseling is a way to kind of cope with that and make good decisions, or the one that's best for you and your family as far as reproducing. That's a very personal thing, and everybody has their own way they want to deal with that. All right, so back to our good friends, the sex chromosomes. Remember, they are the 23rd pair, so the last pair of chromosomes. They are going to determine if somebody is male or female. Just a reminder, males have two X chromosomes. Females have two X chromosomes, males have an X and a Y. Mom can only pass on an X because she has two copies of the X chromosomes. Dad passes on either an X or a Y. So the father's sperm is what determines the sex. Does that sperm cell carry an X, or does that sperm cell carry a Y? They are, actually there's different weights between these two sperms also. They weigh differently, interestingly enough. But so whichever sperm reaches that egg, whether it's a X or a Y, that's going to determine the sex of the baby. Many cultures believe that the female is responsible for the sex of the child, and that women throughout history have been blamed, put to death, divorced, everything else for not producing sons. When actually, guys, it's on you, man. It's not our fault. It's up to you what sex we're producing here. So just a little note. And once again, there are many cultures throughout the world that still hold on to that. Certain gender roles are expected within the societies that are very different than Western societies. And by not having sons, you can be divorced and, basically pushed out if you don't produce sons for somebody. And it is thought to be the woman's fault in a lot of cultures. Sons traditionally weigh more in certain cultures. Okay. And that's the cultural bias towards males is diminishing. However, and I think I have my notes here, basically in certain cultures, like some of the Asian cultures, there is an imbalance in how many men and women there are now because there has been such a strong preference for male children. A lot of the females were given up for adoption or aborted. They have sex-selected abortion so that they are not even going full term with a female. They want males. And now some of those societies who have had these cultural biases towards males are seeing that, oh no, we don't have enough females now. We don't have enough women in our society anymore. And so some of those ideas are diminishing. Now, being female can be quite good in certain circumstances because there are certain disorders that by being female, you are somewhat protected from. And we're going to talk about hemophilia now for that example. These things are called X-linked recessive disorders, and males are more vulnerable to those. All right, our first example of this, and this is often they can call them also sex-linked hormone or sex-linked disorders. And this is particularly a X-linked disorder. And there are other disorders that are known to occur on the X chromosome. This particular disorder, hemophilia, is linked to the X chromosome. The hemophilia is a disorder where you do not clot your blood properly. So if you have a surgery or if you have a cut on your arm, it can continue to bleed and not clot and can cause serious medical emergency because you can actually bleed to death. I actually have a female cousin that has hemophilia. Until then, I didn't know we had hemophilia in our family until she came up with hemophilia. I knew her dad had it, but I didn't know she did. And we're going to see how that happens. Example 1 for hemophilia. Mom has one normal gene right here, that's this guy, and one with a hemophilia variant. So there's a gene along this X chromosome somewhere for hemophilia. This X chromosome, the genes along that X chromosome are just fine. Nothing funky, okay? And then here's dad. Dad, I see it's broken up like that. Ooh, look weird. Dad does not have hemophilia. He has a normal X and a normal Y. How does someone get hemophilia? Well, we'll see. So this child, once again, we have the X with the hemophilia variant on it, and then this X with is a normal X. So this female, she does not have hemophilia, although she does carry that variant for hemophilia. The reason she does not have hemophilia is because she also has a normal X chromosome. Having two X's protects you. So if one of my X chromosomes has something funky going on, my other X chromosome can be dominant over that. And I will not express hemophilia. So that's how this works. This child got two normal genes, no hemophilia here. It's not passed on, and they don't have it. Oops, I have SCA on here. Sorry about that, guys. That's supposed to be hemophilia. Kind of copied some graphs here, charts. So this child here got this Y, a normal Y from dad, which means that this child is a male because we have a Y, so that means it's a male. And this child received the hemophilia gene, the variant of the gene that has hemophilia, from mom. This child will have hemophilia. He has hemophilia because he only has one X. So now that he has only one X, this hemophilia is going to express. So he does have the disease. He will also pass this on to all of his daughters. Because remember, he's either going to, this guy here, this kid is either going to pass on a Y or an X. His sons will be fine. And then the daughters will carry the gene, just like mom does up here. This child received an X from mom that did not have the variant and a Y from dad. So this is a male child again, but they don't have the gene and they don't have hemophilia. Our second, oh, still have SCA on there. Sorry guys about that little error. So this particular example, same thing. Mom has the gene for hemophilia and a normal gene here, the normal one here. Dad has hemophilia. So he has his X gene has that variant for hemophilia. So he has the X here and the Y here. So if he creates a daughter and this daughter inherits this X from mom that has the hemophilia variant and this X from dad that has the hemophilia variant, this daughter is going to have hemophilia. And she is going to pass on the gene for hemophilia to all of her children because both of her X's, her X chromosomes have that deviation on it or have that variant on there. So this next child, we have our hemophilia. This one is a boy because we have Y and X, got the Y from dad, so that makes it a male. The X from mom, unfortunately, there's only one X, so this male is going to have hemophilia. I'm gonna pass it on to all the daughters because this X will go to all the daughters, so every daughter is going to carry that genetic variant. And this child, this is a male child, did not get the X with the variant, did not get the Y with the variant. So this child does not have hemophilia and doesn't pass it on, okay? So those are our examples of sex-linked disorders, genetic disorders, okay? All right, I would ask if you have any questions, but unfortunately, you'll have to email me because I am talking to my computer and I really miss you guys and I miss seeing your faces in class. Okay, genes in the environment is our next section. So as you probably have seen in your other classes, there is something called the nature-nurture debate. If you take an intro, we talked a lot about that in the intro class. If you did not take that and you don't have really any idea what this is, the nature and nurture debate is basically the argument of how much of who you are is due to the genetics that you have and how much are due to your environment. So nature is genes, talking about your genetic makeup. How much of your behavior and who you are is due to the genes that you express, your genetic makeup, and how much is due to your environment, which is the nurture part. So nature is genes, nurture is environment. Scholars basically agree that both play a role, and we have a lot of scientific evidence to show that nature and nurture work together to produce us, to make us into the people that we are today, because we're so awesome. Okay. There is a field of psychology called behavioral genetics. Well, it's kind of a crossover field where you're looking kind of at biology, genetics, psychology, but it's behavioral genetics. This is a particular field that examines the relationship between genes and environment. Behavioral genetics estimates influence of genes and environment on development. So basically, they take a look at genes and your environment, and they try to say, okay, this percentage of your behavior is due to your genes, this percentage is due to your environment that you were raised in. One of the things that behavioral geneticists love to look at are twin studies. Something throughout all of psychology, you will see lots of twin studies because twin studies give you that opportunity to look at people who are genetically similar, and if they have been adopted, that's even better because then they're in a different environment. So a lot of our information comes from twin studies, and these twin studies look at either monozygotic twins or dizygotic twins. A monozygotic twin is, they have exactly 100% the same genotype. They look identical with their genotype. What happens is a sperm and an egg fertilize and a zygote is formed. That cell will divide and you have two genetically identical embryos that form. So basically it's from one cell being split into two and making two separate individuals that are 100% genetically identical. So these monozygotic twins are identical twins. Dizygotic twins are different. Dizygotic twins is when the woman ovulates and there are two separate ova, two different eggs that are released at the same time, and two different sperm fertilize them. So we got a different sperm for this one, different sperm from this one. These twins are dizygotic twins, and these type of twins are no more similar than any other siblings. They just happen to be being incubated at the same time, basically. Okay, so here's monozygotic twins. We call those identical twins. And the other ones we call fraternal twins. I don't have a picture of any of those, but you can have, they can look totally different from each other. All right, but these guys that you're looking at here, they have the exact 100% same genotype. So let's hope one doesn't commit a crime and they're going with DNA evidence, because there could be problems here. Okay. This is a continuation of genes and environment, and we're going to talk about something called heritability. Heritability is the concept where they are estimating the extent of which genes are responsible for differences among people. So heritability is how much have I inherited genetically that makes me different. What's the percentage of that? The range is from 0 to 1. Things that are heritable, basically kind of inherited traits genetically, are intelligence. And intelligence, we can say, is about 0.50, like half based on genetics, half based on environment. An example I like to use here is like Mozart, or with his musical intelligence, or Einstein. Say Einstein never learned to read. If Einstein never learned to read, he probably couldn't have come up with all these theories and all these wonderful ideas that he had. His intelligence maybe could be measured differently, but his overall intelligence, even though the genetic potential was there, he would not have made these discoveries and all of these wonderfully intelligent things that he's done. So basically 50% was, okay, he had a good brain, he had inherited traits that would make him more readily able to get this information and do something with it. And then 1/2 was due to environment. So he was able to read, he had access to information, he had access to libraries and knowledge that he could work with. If you don't have that in your environment, then your intelligence score is going to hurt. Personality, they say there's like a 40 to 50% heritability score for that. They've looked at twin studies where they have seen that, okay, so one twin is like super tidy, and tidiness is one of those things that is heritable. So you have two twins that are separated at birth, adopted by two different families, okay? Twin one says, I am really tidy. I am tidy because my mom taught me that I need to be tidy, and that tidiness is, helps your life run better and everything, and you just have to be tidy. The other twin says, wow, you know what? I am super tidy, but that's because my mom is disorganized. My mom is scatterbrained. Her stuff's all over the place, kind of like my house, all over the place. And so I have to be tidy just in self-defense against her. So we see different things like that, where twins have been separated and they have amazingly similar traits and characteristics. And if you have a chance to look at the Minnesota twin study, I'll put a link in our files for you guys. I'll find somewhere to post that up on Canvas for you guys so you can take a look at some of the results from that study, because it's pretty fascinating to see how twins who are even raised apart, certain traits that they have are very, very similar. political affiliation, religiosity. There's a lot of things that have some type of heritability to them, regardless of environment that they're raised in. Your book has a really good example in there as well, about twins that were separated. One became a Nazi, one was, the other one was raised in a Jewish home, and this was all during World War II. So it was very interesting. The last note on this slide is socioeconomic status, SES. Socioeconomic status has a definite and distinct impact on development. Just like that example that I gave you for Einstein, say we know somebody's got the brain in there, they've got the genetic inheritance to where they could excel academically, but if they are not given that opportunity, and what we'll see throughout our book as we go forward, we're going to see that socioeconomic status, especially here in the United States, when we're looking at it, is going to make a big difference on academic achievement. Because if a child is not given that kind of nurturing environment, access to books, games, parent doesn't have time to do a lot with them, they're busy working and trying to feed their family and make sure they have shoes, they may not have a lot of the extra time and money to do some of the other things that a higher socioeconomic status person would be able to do. And we see differences in language development and everything else that are definitely impacted due to socioeconomic status. Concordance rates. That's the percentage that indicates the degree of similarity in phenotype among pairs of family members. So when we take a look at say we want to take a look at a particular family, and we're going to look at that family and we're going to see what things are expressed, what kind of genetic things are expressed, the phenotype, what can we see that are similarities and differences among these family members. One of the things that we take a look at with concordance rates is concordance rates. There's a lot of research about schizophrenia. Schizophrenia is a mental health condition where a person can suffer from hallucinations, visual, auditory hallucinations, and can have very delusional thinking. And schizophrenia is something that we know is somewhat heritable. We know that there is a genetic predisposition to schizophrenia. We know that if one monozygotic twin, those are identical twins, if one has schizophrenia, the other twin has a 50% shot at developing schizophrenia. We know that with dizygotic twins or regular siblings, there is only an 18% chance, 18% concordance rate, that this child will have, if one twin has schizophrenia, that the other one will have schizophrenia. I put a couple of links to a couple of really good studies down below that show that these are adoption studies with kids with schizophrenia. They found that when kids have a very similar degree of concordance, like if you have twins that only have the 18% chance of developing schizophrenia, if they are in a home that is kind of a less nurturing and stressful environment, they're more likely to develop schizophrenia than kids that are put in a home that is low stress and that has a better family dynamic. But anyway, those two studies talk about that. We're not going to take time to do it here just because I need to get through these videos and try to get us on track here. Gene-environment interactions, epigenesis, and reaction ranges. So genetic activity responds constantly to environmental influence. Once again, our genes are going to tell us to express certain things. For example, height. If I have a gene that would make me tall and I am malnourished and I'm not able to get enough nutrition and I'm in a very bad environment, I'm probably not going to get as tall as I would if I were in an environment where I could have all the nutrition I needed and everything I needed to help me grow to my maximum height. That's a way that the genes and environment can interact. That's an example of it. The gene has a range of expression. So this reaction range that we're talking about here, this is the range from which, like, say maybe my range is going to be from 5'6" to 6'2". This is the top out height for my genes. So according to my genetic makeup, I have the ability to be 6'2" tall if everything goes right in my environment. Maybe I can be 6'2". But if everything doesn't go right, I'm going to be on the shorter side. So that's one way to kind of think about the relationship between genes and environment. And a reaction range is just that range in which a gene can express. So genes establish a reaction range for height, and environment determines where a person's height falls within that range. Pictured here are sisters of the Hammer tribe in Ethiopia, a tribe known for being exceptionally tall. However, if one of these children were deprived from nutrition that they needed and were in a very poor environment for growth, they would not reach their maximum potential. All right, this is the last thing. These are just 3 theories of genotype your book covers, so we're going to cover them here. These have to do with environment and gene interaction. The first one is called passive genotype. This results from the fact that in a biological family, parents provide both genes and environment to their children. So a passive genotype is saying that the way that the environment and genes interact is in a passive way, because I've got these particular genes and my parents, who I share genetic inheritance with, who, you know, they've passed their genes on to me. They're giving me genes and environment. The other 2 types are evocative and active. Evocative genotype says results when a person's inherited characteristics evoke responses from others in the environment. For example, if I am super high, say I'm a kid that bounces off the wall and my mom has anxiety, okay? I'm going to, my behavior, because of my genotype that is, I've got some gene that's making me bounce around all over the place and be really high energy, and my mom's really anxious, that's going to affect my environment. It's going to affect a response from other people in my environment. And depending on what environment I'm in, I may be yelled at, or, you know, told to settle down, or maybe have a more nurturing approach, but my behavior is definitely going to impact my environment, and the way people respond is going to be impacted by the way I'm acting, and that's evocative. The last one is active genotype, environment effects. So this results when people seek out environments that correspond to their genotype characteristics. For example, if I hated to read, hated books, hated to do anything academic, I would probably not want to work in academia, or I wouldn't want to go to college. I would try to find some other environment that suits me better. You know, maybe I'd want to be a dog groomer. And that's what I want to do. I want to groom dogs, but I don't necessarily want to read about it, but I want to, I mean, there are dog groomers that are very educated. I'm not saying they're not, but what I'm saying is that might be something I want to do instead of go to college. So I'm going to seek out an environment that corresponds with my genotype characteristics. So those are those 3 theories. I'm not going to go into a lot of detail about those because I don't think those are as important as the other stuff. And this stuff we're not going to come back to as much. We will see it in the book occasionally, but I'm not very focused on that. All right, genes and individual development, human development. When does individual development begin? Well, the sperm and the ovum go through developmental processes before they are joined. So basically all of this stuff takes place early on. So for normal cells, so in order to create these gametes, these sex cells that we have to have in order to in order to reproduce, our cells have to go through a particular process. Now, for normal cells to replicate, they go through something called mitosis. where a cell divides, it copies all the genetic material, and then it separates and divides, and each part of that cell has a full packet of genetic material. Okay, that's how normal mitosis works in a nutshell. Your gametes, which are the sperm and the ovum, they work a little bit differently. And their process that they go through is called meiosis. So it's similar to mitosis, but it's called meiosis. And there's like another couple of steps involved. So we start off with the chromosome pairs splitting into single chromosomes. So instead of being into those pairs, they're going to split apart. And then after they split apart, they're going to replicate, meaning they're going to reproduce themselves. So each one of these chromosomes is going to reproduce themselves. So after they reproduce themselves, what would happen normally is the cell would split and you'd have two complete packets to go each way in your normal cells. But because these sex cells are a little bit different, we have something occur that's called crossing over. And crossing over means that the chromosomes swap sections of DNA. If you ever wondered why two kids from the same family don't, some of them don't look anything alike, this is why. Because those cells, we have our chromosomes replicated here, and then during the next section, there's something called crossing over, where it's like everything's kind of stirred up and new random pairs are put together. So that's the simplest way to explain it, is that a genuine, new, unique packet of cells or packet of chromosomes were created. And then when this happens, the cell, once again, these are going to replicate and the cell is going to divide. And then the pairs separate again and the cells divide again. So basically, this ends up going from having this 46 chromosomes here to where each of these guys only has 23. Or no, these each have 46. I'm sorry, these still have 46. But then when they break apart again, only half of the genetic material is going into each of these cells. So these bottom line cells are going to be just 23 chromosomes. And if this was a female, if this was the ova being created, then you would end up with 3 cells called polar bodies that are not going to turn into viable eggs and 1 viable egg. Whereas if it's a male, you would end up with 4 perfectly good sperm. But this one might be female, these might all be male. I mean, it could be just kind of a random pairing of genetic material. Okay. So, and I didn't spend a lot of time on that. I just wanted to kind of give you a brief overview on that. So gametes, once again, gametes are our sex cells. They are the sperm and the ovum. And the ovum, once again, are the eggs. Each contain 23 chromosomes. I said this a bunch of times. That could be important. You may need to know that. If I'm saying stuff over and over again, you probably need to know it. And then meiosis is what the name is of the cell division process that creates the gametes. A zygote is what happens when one of these sperms penetrates the wall of this egg. This sperm will penetrate, the tail will break off, the head of this will head to the center of this cell, which is the egg, that ovum. The tail will break off and stay out here, and then this egg will release a substance to prevent any other sperm from penetrating in. So this little guy is going to carry his 23 chromosomes. This big cell already has 23 chromosomes. So when they pair up, we get our 46 chromosomes. And then this is what you call a zygote. As soon as that sperm enters that cell, that is a zygote. When that genetic material bonds together, then we are looking at a zygote. Okay, more information about gametes. So the sperm, there are millions produced daily starting at puberty. So when a male hits puberty, they start producing sperm from their testes millions daily are produced. There are about 100 to 300 million released per typical ejaculation for a male. And with age, the number of sperm and the quality of sperm decline. So, you know, they're not going to swim so straight. They might swim a little crooked, have a little twitch. They just aren't going to do as well with age. The ova are very interesting. Females, basically, when they are in utero, so when I am developing inside of my mother's uterus, I am developing all of the eggs I will ever have in my lifetime. So they are produced while female is in utero, that's developing in the uterus. We are born with about 2 million ova, which are these little egg cells, right? By the time we hit puberty, we're down to about 300,000 of these little critters. And then we only have about 400 that will mature. And most women run out of their ova or eggs in their 40s. So it's pretty interesting. The last portion of meiosis to occur. So these eggs kind of sit there in limbo until we reach puberty and until we are in a reproductive age before we start completing the whole process. So women run out in their 40s. All right, that was the end of this first video, part 1 for chapter 2. You guys take care. I hope to see you soon and have a lovely afternoon. Now I have to figure out how to stop recording. Okay, take care. Really?